Hello everyone, welcome back to my channel. Today we'll be again taking up one of the biomolecules, that is amino acid and proteins. Basically the biomolecules which we'll be talking about is proteins, but protein is related to amino acids. All proteins are made up of amino acids. Amino acids are nothing but the building block of proteins. So in this particular video, we'll be first talking about amino acids, and then we'll be going up with the very important biomolecules, that is nothing but the proteins. Now, talking about proteins, okay, what do we know about proteins? Okay, protein is a very essential biomolecules, okay, very essential for human beings, not only for human beings but for all living organisms. Okay, the chief source of proteins which we take in our day-to-day life is milk, cheese, pulses, peanuts, fish, meat. These are all the various source of proteins which we take in our day-to-day life. Remember that this particular proteins are nothing but chemicals only, nothing but high molecular mass, complex biopolymers, which we say, okay, of amino acid. As I have told you, amino acids are building block of this particular protein, okay, and which is very much essential for the survival of living organisms. Okay, the word protein, okay, if you talk about this particular word, this is actually a Greek word, proteus. Okay, now what do you mean by proteus? It's a Greek word for prime importance. Okay, this was found out, this particular biopolymer was found out to be a very vital chemical substance essential for growth and maintenance of life, okay, without which we cannot survive, which the life or cannot be maintained. Okay, if you see protein in our body or in any living organisms, almost the 20% of protoplasm is made up of in plants or animal cells are made up of proteins. Okay, in humans, if you try to talk about where actually proteins are present in our muscles. Okay, the framework, okay, our framework itself is protein. Skin, okay, the hair, the nails, tendons, arteries, connective tissues, whatnot. Okay, all this are nothing but proteins. So in short, what I can say is that each living cell is made up of thousands of different kinds of proteins. Okay, that is what actually protein is so important for us, and that is what we are planning to do today. Okay, try to understand the importance and basically try to understand the chemistry behind it, why actually we or how this particular protein forms in our body, how amino acid is essential for the formation of proteins, and how actually this protein gets different structures. Okay, and how we can study this different structures of protein. So that is what we'll be focusing more on in our today's video. Okay, so let us go ahead with our video today, or for our lecture today, that is on amino acid and proteins. Okay, so coming for the introduction part, proteins are constituents or constitute an important class of biomolecule, as I have told you, essential for living organisms. Okay, they are 19% of an average human body actually constitute the proteins. Okay, and so several vital functions, okay, in our body. The word protein, as I have told you, is derived from Greek word, means actually Greek word proteus, which means of prime importance, as I've already told you. They are polymers, they are biopolymers. Okay, they are naturally made. Okay, and remember that what they are made up of is nothing but amino acid, and be very specific, it is alpha amino acid which makes the protein in our body. Okay, so we'll try to see that how this alpha amino acid basically joined together to give you a polymer, which is nothing but a biopolymer, which we called as protein. So how this reaction happens in our body and how this reaction leads to the formation of protein. Okay, that is what we are interested to know. So thus proteins are nothing but polymers of alpha amino acids. Okay, so as I have told you, before going to proteins, let us first try to discuss what are alpha amino acids. So amino acids. Amino acids are the basic structural unit of proteins. Okay, so whenever we talk about amino acid are basic structural units of protein. That is what protein is made up of. So when we say amino acid, by the name itself, aminos actually goes for NH2 group, and acid is nothing but carboxylic acid. Okay, so it is a compound which contains these two groups, that is carboxyl group and the amino group, as you can see in the screen. Okay, and why do we call it alpha amino acid? Obviously, our carboxylic acid is the functional group over here. This functional group is attached to a carbon, which you can see at the center, and this carbon is the alpha carbon atom, right? Alpha carbon atom is nothing but the carbon atom which is attached to the functional group, and this alpha carbon, okay, which you can see, this alpha carbon is attached to NH2 group. That's why we call it alpha amino acid. So depending upon to which carbon the amino acid is linked to, we can have beta amino acid or gamma amino acid. But remember that what makes us, okay, what are the proteins in our body are all made up of this particular molecule. So basically our structure is basically formed of this particular molecule, whether we call it muscles or whether we call it amino acid, whether we call it our hair or nails, is basically this particular chemical having different structure, forming in different ways, having different sequence, and giving you different class of proteins. Okay, so that is the structure of general structure of an alpha amino acid. And this R, okay, which you can see, that is what we call as the side chain, and that varies. Okay, and as it varies, we get different types of amino acid. In general, we have nearly 20 amino acids in our body, which makes actually different kinds of proteins in our body. Okay, we'll be trying to see the structures of all those as we go ahead in this lecture. So that's what I have told you, okay, that this particular amino acid has a acidic group as well as a basic group, a carboxylic group, which is acidic, amino group, which is basic. So what basically happens is when you have such compounds where you have an acidic as well as basic group, then obviously acidic group will transfer the H plus very easily. So carboxylic acid will give you H plus very easily. That H plus will be taken up by the basic group, that is NH2 group, forming NH3. Because obviously NH2 will have a lone pair of electron through which it can easily form a bond with the H plus. As a result, what will happen is the amino acid will not exist in this form, but exist in this ionic form, where the carboxylic group will be forming carboxylate ion, you can see O minus, and your amino group, okay, will be forming NH3 plus ion. Okay, as a result of which, what will happen that in the same compound you will have a positive charge and you will have a negative charge. Such class of ions are known as zwitter ions. And remember that whenever we talk about this zwitter ions are nothing but neutral species. They are neutral. Okay, even though they have charge, but they are neutral, but they have positive charge as well as negative charge in the same ion. So altogether they are neutral, no doubt in that, because the positive neutralize the negative, but they have the two polar, actually polar groups, okay, positive and the negative. That is what we call as dipolar ions also. Okay, you can clearly see that is nothing but zwitter ion. So remember that the amino acid in our body exists in the form of zwitter ion rather than the normal neutral form. Okay, but remember that as a whole, this zwitter ion is neutral, it's not charged. Okay, if actually kept in an if you try to take this particular zwitter ion, it will never be a charge system, it will always be actually a neutral system. Okay, then coming for the structure of alpha amino acids. Okay, the different alpha amino acid which is present in our body. There are 20 amino acids, as I have told you, and these are the different structures of those 20 amino acids. So these are the 20 amino acids which we have in our body: glycine, alanine, serine, threonine, cysteine, valine, leucine, isoleucine, methionine, proline, okay, phenylalanine, tyrosine, tryptophan, okay, aspartic acid, glutamic acid, asparagine, glutamine, histidine, lysine, and arginine. Okay, these are the 20 amino acids which forms the various proteins in our body. Okay, now the question is how they do that. But before going into that, let us go into the classification of amino acid. How do we classify these different amino acids? We have nearly 20 amino acids in our body. So how do we classify them? Okay, one of the classification of amino acid is based on its essentially whether it is essential or non-essential. Okay, so how do we know that which amino acid is essential for us and which amino acid is not essential? It is actually based on whether our body can produce it or not. If our body can produce a specific amino acid, that means we need not take it in our regular diet. Okay, so in our diet, if we skip it also, our body can produce it. Our body has a mechanism to make those amino acids. Those amino acids are said to be non-essential, which we don't require because our body can make it. Our body has a mechanism to synthesize those amino acids in actually our body. But there are many other amino acids which cannot be synthesized by our body. Okay, such amino acids are nothing but the essential amino acid which must be taken in our diet regularly. Okay, absence of which or deficiency of which causes various problems. Okay, so that is the definition of essential and non-essential amino acids. So as I just read it on, the essential amino acids are those amino acids which the body cannot synthesize, are called essential amino acid, and the non-essentials are those which the body can synthesize, which we need not take in our regular diet. Okay, that is how amino acids are classified into the essential and non-essential. The list of essential and non-essential amino acid you can see in your screen. Then coming for the next, that is the neutral, basic, and acidic amino acid. So how do I know that which amino acid is neutral, which amino acid is basic, and which amino acid is acidic? Okay, so the neutral amino acid, as I have just told you, that whenever we talk about amino acid, it has two groups. One is amino and one is acidic. Okay, so one is acidic group, one is basic group. So whenever you have the equal number of acidic and basic group, means one amino group and one carboxylic group, then they neutralize one another, and such amino acids are nothing but neutral amino acids. But there are amino acids which have two amino group and one carboxyl group. So that means your basic group is more and acidic group is less. Such amino acids are nothing but basic amino acid, which you can see in your screen, the example of which is lysine, arginine, or histidine. If you talk about acidic amino acid, it's just the opposite. You have more of carboxylic group and less of amine group. That means you have two carboxyl group and one amine group. Example, aspartic acid, asparagine, or glutamic acid. Okay, so if you see this structure, you'll find out that there are two carboxylic group in this particular amino acid. So that is how they are classified. Okay, that is the classification of amino acid. Coming for the next, now what we'll be going to do is peptides. Okay, so what are peptides? Basically, when two amino acids join together, okay, in the process of forming protein. So the product which you get when two amino acids react with one another is nothing but peptide. Now, depending upon how many amino acids are reacting, 1, 2 amino acid, 3 amino acid, 10 amino acid, depending upon them, we categorize in different kinds of peptides. Like if 2 amino acid will react, we call it dipeptide. If 3 will do it, then we call it tripeptide. If 4 will do it, we call it tetrapeptide. Usually from 2 to 10, we call them oligopeptides, and more than 10, we call them polypeptides. So let us try to see now that how it actually happens, how in our body, when actually amino acid is present, then this amino acid react to make what we are, okay, the proteins. So as I have told you that whenever we talk about peptides, peptides are amides, remember that. Okay, so how it is formed? It is formed by condensation reaction of one alpha amino acid or one amino group of alpha amino acid with the carboxylic group of the another molecule. Okay, always remember that whenever an amine and a carboxylic group will react, what they give you is another class of compound which we call as amide. That's why the proteins are also known as polyamides, which form many amide linkages. Just to make it very clear to you, let me just show you this particular reaction. You can see that we have glycine. Okay, that is the simplest amino acid which we have. Okay, so this particular glycine actually reacts with another amino acid which is alanine. Okay, I hope you can see clearly over there that you have a carboxyl group, okay, COOH group, and you have a NH2 group. So there is a condensation reaction now. So what is basically a condensation reaction? Where two molecules join by releasing small molecules like water or HCl or NH3, depending upon the system. Like over here, the small molecule which is released in the process, elimination of molecule of water is taking place over here. So how that water is eliminated is basically this OH group of carboxylic group and hydrogen of this amine group will go together as H2O. Okay, and what happens is this two, the CO and the NH join together. So this is nothing but an amide linkage, what we called as a peptide bond. Okay, so whenever we talk about proteins, the very common word which we always talk about is proteins are linked with one another. The amino acid in proteins are formed by peptide bonds, right? What is a peptide bond? It's basically an amide linkage, where a amine group and a carboxylic group react with one another. And when they do so, what will happen is they release water molecule. And that water molecule, which is released in the process, joins this two. Okay, so once that water molecule is released, this two part of this amino acid join together by an amide linkage, which you can clearly see on the screen marked with red. And that bond, we call it as peptide bond instead of calling it a amide bond or amide linkage. Okay, and since it is form of glycine and alanine, so what do we call it? Glycylalanine. Okay, so GLY-ALA. The amino acids are usually written in the form of these three words, their first three letters, okay, of the name. GLY stands for glycine and ALA stands for alanine. Okay, and the name of this particular dipeptide, since it is formed of two peptide, is nothing but the glycylalanine. Now, one very interesting thing over here you need to understand is, once this forms a dipeptide, can you see again there is a NH2 group and again there is a COOH group. So what will happen? This NH2 group can react with another COOH group of another amino acid, and this COOH group can react with another NH2 of another amino acid. Then again you will have one COOH group, one NH2 group. So this will go on continuing. So it will not just stop with two molecules. It can just go on and forming a long chain. That is what we call as a polymer formation. Okay, that's why we call it biopolymer. So it will just go on and go on and go on and forming long chain. Okay, one end will have actually carboxyl group and another end will always have an amine group. That is what we call a C terminal and N terminal, okay, of a protein or a polypeptide. Okay, so as it goes on, if it forms three amino acids are involved, we call it tripeptide. If 4 tetrapeptide, 2 to 10, as I've already told you, will be known as oligopeptide. And if more than that, then we call it polypeptides. Okay, so that is how actually it will go on. Okay, and that is how actually we can understand that how amino acid actually forms the protein. Okay, why amino acid is important. Okay, because those amino acid actually forms or linked together to give you that long chain, and that long chain is nothing but protein. Now, how this protein takes up the shapes, different shapes, right? Our hair, skin, muscles, hormones, different proteins are there. Okay, now how this different proteins takes different shapes. Okay, that is what we'll be trying to understand next. So that is what we are trying to understand now, that is the structure of protein. Okay, so whenever we talk about the structure of protein, obviously protein is a very complex biopolymer or very complex system. We cannot just study its structure in one go. So that's why the structure of protein is studied in 4 different levels. We call it primary, secondary, tertiary, and quaternary. Okay, so let us try to see in this 4 different levels that how do we understand how actually this particular chemicals, okay, which are formed in our body takes the shape of actually what we can see ourselves as, our hairs or nails or skin or the muscles. Okay, how it goes for that. Okay, so that is what we are trying to understand. The first structure of protein, what we'll be trying to understand is the primary structure. Okay, so what do you mean by primary structure? Basically what happens is whenever a particular protein is formed, it has a particular sequence of amino acid. An amino acid, let's say if I write as A, B, C, D, 4 amino acid. So in a particular protein, first A may react with B, B may react with C, C may react with D. In some other protein, C may react with A may react with D, D may react with E. Let's say some another amino acid. So imagine that you have 20 amino acids. So how many permutation combination you can do and how many different kinds of proteins you can have in your body. Okay, so that is what we are trying to understand. So this particular sequence of this particular amino acid will decide what kind of functions this particular proteins will be going to do in our body. So each particular sequence decide the importance of a particular protein. Okay, your hair is different from your nails is different from your muscles is different from the hormones. So all these differences comes basically from the primary structure, the sequence of the amino acid. Okay, now as I have already told you that any particular amino acid primary structure or the amino acid will have two terminal, the N terminal and the C terminal. Basically, it is the amino group and the carboxylic group. So if I just read it out, the primary structure is represented by the beginning of an amino acid whose amino group is free. That's what I've told you. Amino group will be free. And in the another end, the carboxyl group will be free, and then this chain will actually go on forming a long chain, or this particular amino acids will go on forming a long chain, and that is how the primary structure is formed. So once this sequence is formed, once the sequence of amino acid is formed, what next? What will happen now? So it will form a long chain. What will happen to this long chain? This long chain goes for two different structures. Okay, that is what we called as actually the secondary structure of protein. Okay, in this secondary structure of protein, so what basically happens in the secondary structure is, in this secondary structure, this long chain, okay, this long polypeptide chain will actually form hydrogen bonding. Okay, that is kind of intermolecular force. So hydrogen bonding with itself or with another protein chain, giving you two different types of structure. The first type, what we are trying to do is the alpha helix structure. Okay, so what is this alpha helix? You can see in your screen, that is what is alpha helix. Okay, this structure is formed when the chain of alpha amino acid coils as a right-handed screw. Okay, if you coil it right-handed as a right-handed screw. Okay, and what will happen is once you actually coil it, can you see in the screen that the particular actually the carbonyl group of the amino acid, okay, when you actually make it a spiral form or an alpha helix form, then what will happen is it will meet in another turn. Okay, so it will turn, right? It will go on turning. So once it will go on turning, what will happen is this turns, okay, will be linked to one another. So who will hold this turn? Someone must hold it if it wants to exist in this way. So something must be there which is holding it, binding it. What is binding it is nothing but this particular carbonyl group, okay, the electronegative oxygen over there, and the NH in another turn, your NH will be there, that hydrogen of that NH, which is electropositive compared to nitrogen. So it will have a positive end over there in hydrogen and negative end in that particular oxygen. So this two will interact with electrostatic force of attraction, and that is nothing but hydrogen bonding. And that hydrogen bonding will hold it and will not let it uncoil. So it will remain in coiled form because of that hydrogen bonding. So hydrogen bonding becomes very, very crucial for secondary structure. Okay, so the basic loss actually of the secondary structure will happen if this hydrogen bonds break. Okay, so this hydrogen bond makes this secondary structure possible. So the alpha helix is basically because of this secondary structure. Okay, and it was found out when it was studied that this hydrogen bond, okay, the hydrogen bonding between different unit is responsible for alpha helix, and the side chain, obviously this amino acid will have this side chain, the R group, as I have told you, and this side chain will remain actually outwards of the coil. So it will not disturb the coiling. Okay, it will remain outwards of this coil. Okay, it will go away. It will not go inner to the coil. Okay, as actually it is formed. Okay, such proteins are actually basically elastic. Okay, and they can be stretched. Okay, and remember that the hydrogen bonds, okay, are reformed on releasing the tension. So when you actually stretch it, some of the hydrogen bond breaks, it becomes weak. And once actually you release that, then they again actually take the original shape. Okay. Wool, hair have the actually this kind of alpha helix structures. Okay. Now coming for the next, that will be the beta pleated sheet structure. Okay. By the name itself, now what will happen is one protein will not coil. Now there will be many proteins, okay, which will be laid on one on top of another or side on sides of one another, and they will be holded to one another by hydrogen bonding. So over here, many chains will be linked to one another by hydrogen bonding, which you can see in your screen now. So what you can see in your screen is, see, this is one chain and this is another chain. Now there are two different chains and they are holded to one another by hydrogen bonding in the very same way with the carbonyl group and the NH group. And this hydrogen bonding holds this chain together and they form a sheet structures. Okay. One of the very common example of this particular type of protein is silk. Okay. Silk has this beta pleated sheet structure. That's why we call it a sheet structure. Okay. It forms a sheet where all this particular proteins or this particular polypeptide will be lined, okay, and actually holded by strong hydrogen bonding. Okay. So that is how actually it gets a secondary structure. So once it gets secondary structure, then what will happen is this particular protein now will go for its tertiary form. Okay. So in its tertiary form, what will happen is, so what basically this tertiary form represents, this tertiary form represents the overall folding, okay, of the secondary structure to give you actually a molecular shape. Okay, what actually it is present in our body, right? How the protein is present in our body is basically present in the tertiary form. Okay, so it folds. Okay, and when it folds, you get two types of molecular shapes. Okay, so when you have amino acid joined together to give you primary. When it coils or forms a sheet, you get secondary. And that secondary, when actually the overall folding will take place of that secondary, how it is existing in our body, that is nothing but the tertiary structure. And that tertiary structure are basically of two types. The overall protein is also sometimes classified as two types of protein. We call it fibrous and globular. We'll be coming into that next. Okay. So remember that tertiary structure is nothing but the overall folding of the secondary structure. Okay. But there are some cases where it goes more than tertiary also. It goes for quaternary structure. So what is this quaternary structure? This quaternary structure happens when your particular protein which you are talking about is made up of more than one unit. Sometimes it happens. Okay. Sometimes the proteins are made up of more than one polypeptide unit. That is what we call as subunits. Okay. So if it happens that the protein which you are talking about is formed of more than one particular amino acid chain or polypeptide chain, then what will happen? All this polypeptide chain will have its primary, secondary, tertiary. Then they coil around themselves. Okay. All these different subunit to give you another structure. That is what we call as quaternary structures. So it happens only for those proteins which have more than one polypeptide chain. There are many proteins which does that. Okay. So you can see on the screen diagrammatically represented primary, one strand, secondary. Okay. Either it will coil or either it will form beta pleated. And this secondary again will go for tertiary. You can see in your screen. Okay. It will coil to give you two shapes, fibrous and globular, which will be actually looking into it next. And then if you have many or two or three polypeptide chain, the subunits, then all of them will coil together to give you the quaternary structure. Okay. So that is how actually we can understand that how these proteins are existing in our body, how it starts from the amino acid and comes to this original form. So as I've told you that there are two types of proteins, basically one is fibrous and one is globular. So let us try to see this. Okay, so basically this is the tertiary form of proteins, okay, how it is existing. So the fibrous form, these are made up of polypeptide chain that run parallel to one another. So what will happen? This polypeptide chain, when they are running parallel to one another, okay, so what will happen is they will be holded by one another with hydrogen bonding, as I have already told you. So what will happen is they will form actually this, what do you call it? This particular proteins, which will be actually lying on one plane. Okay. And they will be kept on top of one another. Okay. So they can be stretched also. Okay. and they can be contracted also. Okay, they are insoluble in water. Very common is the alpha keratin, okay, which we have in our hair. It's not soluble in water. Okay, wool, silk, nails, all this, myosins, muscles, okay, all your muscles basically are formed of this class of proteins, okay, where the polypeptide chain run parallel to one another. They are just stacked on one on top of another, and they are holded together by this particular hydrogen bond, or we call it disulfide bonds, okay, which holds them together very properly. Coming for the second type, as I've told you, which we called as globular, most of this are nothing but the hormones, the hemoglobin, which are present in our body, insulin, okay, all this actually are soluble in water, okay. So what they do is they have more or less a spherical shape, okay, where they actually don't go for a stretched shape, okay, where they don't get stretched. Okay, they don't like straight on one on another and one on top of another. What they basically do is they form a globular or a spherical shape. Okay, and remember that usually the alpha helices are tightly held, okay, by weak weak attractive forces of various type, talking about hydrogen bonding or disulfide linkages. Okay, and what will happen is this particular system, this particular proteins are usually soluble in water. Okay, and the common examples of this kind of proteins are insulin, pepsin, hemoglobin, cytochrome, okay, albumins. All these are nothing but proteins only, okay, but they are not actually visible, okay. Obviously, they are dissolved in the system in water, which we have in our body. And why they are soluble? Because they form globular protein instead of fibrous proteins, okay. So this is how actually we can classify the two tertiary structure of proteins into two different types of proteins. One is fibrous, okay, which you can see, okay, and the globular, which are usually soluble, okay, hormones and hemoglobin and the cytochromes and the pepsins, okay, all these different proteins, okay, which are soluble in our system. Now, coming for the final topic of protein, that is denaturation of protein. Now, I hope every one of us once have actually experienced this, that whenever we boil an egg, it actually coagulates, okay. So whenever actually there is any protein, okay, remember that in its native form. What do you mean by native form? That means how it is present in biological system. That particular protein is known as native protein, okay. So whenever we talk about native protein, remember that native proteins are those proteins, okay, where actually it has a specific three-dimensional structure, the tertiary or the quaternary structure, and with a specific biological function. They are native protein. But this native protein, okay, if you bring out of the system, okay, like if you get a cut, okay, some actually hemoglobin will come out, right, along with the blood, right, the plasma, some proteins will come out. If you actually boil an egg, what will happen? It will coagulate. What basically you are doing is you are giving it actually a what do you call it, a change in the physical or the chemical nature of it, okay, what it was not in that particular environment. It was not in that physical environment or in chemical environment, what it was in the biological system, because of which what will happen is this hydrogen bonds, which we have seen, they tend to actually break, and the globular proteins tends to actually open up, or the alpha helices which you will have will start uncoiling, and they start breaking, and they lose their functions. Remember that such type of proteins, okay, when the protein does that, okay, when they are subjected to such kind of change in pressure, temperature, chemical change, pH, okay, when when they are not in the same environment, then they lose their function, they lose their tertiary structure, they lose their secondary structure, they lose their quaternary structures, okay, and lose their function. And such actually process, okay, that phenomenon is known as denaturation of protein, okay. Very common, as I have told you as examples, okay. That is nothing but the what do you call it, the coagulation of egg, okay. You have the curdling of milk, okay. All these are examples of denaturation of protein. And very important, remember that whenever denaturation takes place, as I have told you, it will lose its structure, it will lose its function, but it will not lose its primary structure. Means the primary structure remains in denaturation process. Remember that the amino acid will still be linked to one another, but yes, lose their secondary and tertiary structure, as a result lose their function. That is how actually we can understand what a protein is. Okay, so that is all about the proteins and amino acids. So thank you, everyone. Do subscribe for this channel if you like the video for explanation on various topics in chemistry. And do follow me on Instagram and Facebook, and like my page and follow it as Kafle Search. Thank you.